Good afternoon, everyone. If there's anyone in the back wants to come on up, we got plenty of seats in the front. I'm Stu Chandler. I'm the CSO of Aclaris, and Aclaris is very happy to be able to sponsor this now meeting for the third year in a row. I think the first one was You Don't Know Jack, which hopefully is not true certainly isn't true for most of the people in this room. Last year, I think we did Jack Be Nimble, and this year we're going to be talking about HiJaKing topical therapies for new indications in dermatology. So the first portion of the session today will be led by Jeff Travers. The session will focus on the specifics and special things you need to know and do in order to effectively develop a topical agent, topical JAK in particular, for dermatologic applications, which is very different from developing systemic drugs. There's different sets of rules you have to follow, and we're going to learn a little bit about those today. And then the second session will be led by Brian Kim, who's going to have quick 7 or 8 minute sessions on new indications for the JAKs, particularly focused in dermatology, though not exclusively. And so let's bring on Dr. Travers. Thanks very much.
Well, thank you very much. And first of all, I have no conflicts of interest. How often I get this question as a pharmacologist and dermatologist: Jeff, I've got this new molecule, but I'd like to test it in mice, and potentially I want to put it into creams, and I want to use it topically. And that is an amazing art, and it fits with our first speaker, Dr. John Jacobson, who is also an artist, and he'll be talking about the art of design of topical drugs. Dr. Jacobson is the Vice President of Chemistry at Confluence Discovery Technologies. So John, you want to come on up?
Okay, thanks, Jeff. So today I want to cover in a very, very basic talk, some of the design principles that are involved in the design of topical drugs. And it will center somewhat on JAKs because that's the nature of this program. So when we think about topical drugs, I first want to segue into really what is medicinal chemistry, because if we don't have a basic understanding of that, the rest won't make too much sense. So medicinal chemistry is really an optimization of a number of features of a molecule. And you initially start off with potency, because you have to be potent at your target in enzyme, cell, and obviously in vivo. But then you need selectivity against off-targets. And if we're dealing with kinases, that's up to a little over 500 off-targets that you could hit. Then you start to deal with the properties that are involved in stability. So you need good solubility to help with absorption. And then you need to have good metabolic stability to make sure that you have distribution and minimal metabolism so your molecule will work. And then if you're successful with all of that, safety becomes important, and at the very end, you need to own it. So an example I'm going to show on the next slide is basically the screening hit that was involved in the discovery of tofacitinib. So basically, chemists in Groton in the late 90s and early 2000s were starting on the JAK program, and they started with a screening hit, 352664. And this hit was reasonable, but it was very lipophilic. It had reasonable potency. And the bottom part of that structure, as you see, has a proloprimidine. And that portion is the portion that kind of binds into a kinase cleft like ATP does and makes key interactions. So they didn't want to modify that very much, but they modified the top set of rings. And usually you don't want that big of a set of rings in that way because it's flat, and it often causes some major issues with chemistry, basically, as far as making it optimal for your purposes. So they cut off one ring, which was the phenyl on the left, and they found out that they actually increased their cell potency. So that's a good thing. And at the time that Flanagan's group in Groton did this, they didn't know how basically how ATP even bound to JAK, let alone their inhibitors. So they were doing this relatively blind, and kind of the way MedChem worked for a long, long time. So they cut off the one ring, then they fragmented the other ring to give it the compound on kind of on the far right of the slide. Basically, this compound then had a similar potency, so they were able to eliminate a little complexity that they didn't want. From there, they explored a fair amount on the center ring, on that pyridine, on that cyclohexane ring, and they turned to a natural product for one set of their analogs, and that was carvone, which gave them that methyl isopropanol compound in the middle, shown here. I don't think the pointer's working. So basically, that provided a much improvement in potency. So they obviously were interacting with the kinase in some unique ways that they didn't expect. So the problem was they had a molecule that was way too lipophilic for an oral discovery program. So they wanted to decrease that, and it was also not a very good compound for synthetic E's, because you can't do a lot with that quickly. So they went and put a nitrogen in the cyclohexane ring, basically to make a pyridine, and from there then they were able to analog. And one early analog is kind of on the bottom left, and that's with the trifluoroamide, basically. And this compound lost a little potency, but it now is somewhat drug-like. It had okay metabolic stability, solubility was okay, it's in the right range. From there, they spent a fair amount of time trying to find something that would replace the CF3. And they found, within the several hundred compounds, they finally found that nitrile was very unique. And that when they had the amide with the methylene nitrile on there, they had a significant boost in potency in enzyme and cell. Their lipophilicity was further lowered. Their metabolic stability was some of the best in that particular series, and that in the end became tofacitinib. So every change they made, basically, and any atom in there impacts a number of properties. And that's kind of the way medchem works. You make a change, some things are good, some things get worse, and some things are not predictable. And that's the way drug discovery is. It involves a lot of intelligence, a lot of luck, unfortunately, too much sometimes. But in the end, you can usually find a way if you're diligent in how you pursue this. So when we think about how we would do drug discovery now, if we were interested in, let's say, a topical JAK inhibitor, we might use structure. So we determined the structure of tofacitinib bound to JAK3 as well as a couple of the other JAKs in around 2008. And one thing we noticed well, first off, you see that the core, the pyridine core, binds to the hinge as shown on the slide and making two hydrogen bonds there. And that's expected, and you probably don't want to change that because that's a very good moiety for this particular type of kinase. And then you see the compound come out underneath, just like ATP would, out towards the phosphorylation sites. And so when we analyzed the structure, we found that the nitrile was pointing up into the G loop, that's that red ribbon on the top of the kinase, and it points up into there into a little pocket. And by doing that, it's actually displacing an unfavorable structural water, which proves enthalpy and gives a significant boost of potency. A second thing that we noticed was that the methyl on tofacitinib projects into the bottom of the pocket or the floor, and that's into a JAK specificity pocket that's fairly unique to JAK kinases, and most kinases don't have that. So this is how tofacitinib works as far as its potency in JAK3. It actually binds in the other JAKs in a very similar way. And so it's a good starting point if you want to think about how to optimize a compound for it, except maybe this isn't your best starting point because this is fully optimized for an oral drug. So when we think about structure, we often draw structure, at least chemists do 2D because that's the easiest way. But really any structure is obviously in 3 dimensions. And any structure, because of rotatable bonds, has many, many, many conformations. It can be up to the thousands. And so you have to understand those conformations, and that's not always so easy. But it's that 3D shape that's actually critical for potency against your target. It's also critical for possibly different 3D shapes to hit other targets. And that's something that you're always wrestling with. So you need to kind of understand the energetics of your shape. And shown on the 3D structure on the left, I'm looking at this, basically on the left, that's tofacitinib in its bound conformation. So it looks very different though in unbound, and in a minimal shape it may be very different yet. And I've indicated in the color the basically polar groups, so the blue and the red are areas of polarity that basically affect the polar surface area and can help with interactions. The areas in the white and gray are just lipophilic. nature's parts of the molecule. And lipophilic interactions are actually more important sometimes than hydrogen bonds because a lipophilic to lipophilic interaction, kind of like grease to grease, is very potent and provides greater energetics than just making a hydrogen bond, and usually less of an issue. And so when you think of a structure, you realize that it can be multiple structures, but the key thing is if you know what your bound structure is supposed to look like, you need to ensure as much as you can through modeling that the energetics are very similar. Because if your bound structure has much higher energetics to get into that conformation, it may look good on paper, but it won't work because it will never get that way in nature. So when you think about structures, we obviously can design far more than we can make. It's just not that easy to make compounds. And so we need to predict things. So the things we care about are showed, that I've already talked about, potency, selectivity, and so forth. We really care about that, and that's what we want to optimize. But you can't predict that as well. What you can predict are a number of the other features. And some are very easy. Molecular weight, hydrogen bond donors and acceptors, a number of those. That's factual. It's really easy, very accurate. Polar surface area is not bad. pKa is a little trickier. Some of the properties like interactions with the target, metabolic soft spots, that comes from knowing drug discovery, software tools we have, and so forth. Those are very important. That's part of the design process. Another part that's actually very, very critical is lipophilicity. And lipophilicity is in a number of predictive models as a part of the core of that prediction. The problem with lipophilicity is there really isn't a great way to predict it. There's a number of models out there. We use several. But in the end, you always want to benchmark to real data. So that's one of the things that you can predict, but it can lead you astray unless you generate data to understand it. And so basically in drug design, you would always try and do a lot of these predictions ahead of time so you know what you're doing and you can make the best progress more quickly. So now when we think of oral versus topical drugs, so I've already discussed properties that we want. When you think of an oral compound, those properties start to differ from a topical compound. So with an oral compound, you want a compound with high potency because it has to get in, it has to be distributed, it has to stay on target long enough. It definitely needs to be pretty potent. It also has to be metabolically stable. It has to go through the liver, so it has to be robust enough not to get degraded from the liver very quickly. It also has to penetrate the GI tract. So those are very critical features. Solubility is quasi-important. It's nice, but many oral drugs aren't very soluble. So those are some of the key features of an oral compound. For a topical compound, they start to differ. Solubility is critical for a topical compound because if you don't have it, it gets to be a lot more difficult. And you want solubility not only in water, but in the excipients you might formulate your drug with. Because if you think about a topical agent, it starts off between a 0.5 and a 2 or a 3% formulation. That's actually very concentrated to put an organic molecule in. So you have to have high solubility. You also have to have high permeability because it has to get across the stratum corneum and other parts of the skin that I'll get to in a second. And that's very critical because if you don't have that solubility and permeability to get in, you won't, or you won't very well. Metabolic stability is one of those optional things. In many targets, it's not a problem, and you wouldn't care. But if you did have concerns over a systemic burden of your topical agent, you might want to have higher instability from metabolism, basically the liver. So now let's think about skin. Drug delivery into skin is still difficult. There's a lot of expertise that's been done in formulation, so it's getting better, but it's still not the easiest thing to do. And if you start with an oral compound that's been designed highly for an oral use, and you're trying to get it into the skin, it can be very difficult because you haven't designed it specifically for what you want to do. The key issue with the skin is you have the stratum corneum, which is very hard to get through, the epidermis and the dermis, and it depends how far you want to go, but if you want to get to the bottom, near the hair follicles, then you've got to go way down. But basically, these different layers work against each other. The same type of molecule, if it's very lipophilic, might be fine for the stratum corneum, but it won't get through the others. So it's a very balanced compound. So we can use our properties to predict, like permeability, solubility, predict or measure. Molecular weight and hydrogen bond donors are critical for a topical agent. For hydrogen bond donors, you want 2 or less if possible. And for molecular weight, 350, 300 would be very good if you can. It's hard to have a potent compound, molecular weight 300, so that's your kind of your caveat. There are also rules, and for oral drugs, there are rules that are quite famous, and they're called Lipinski's rules that helps you predict if you're going to have oral bioavailability. These rules somewhat apply to a topical agent, but they're more restricted. And there's a little shift in how they balance, but there are rules, and Troy has worked out those rules for us, and they're quite useful and applicable towards topical drug discovery as well as other methods of dosing. There are also ways to do this mathematically, and Guy and Potts have worked out several equations, and one is basically measuring Jmax, which is the rate of flux through the skin. And basically, Jmax is really just a permeability constant that you calculate times concentration, so it's relatively simple. The permeability constant, however, is shown below, and it's really basically a comparison of log P and molecular weight, but in a non-aligned way. So basically, molecular weight and lipophilicity work against each other. So you can't go too far one way or too far the other way, or you'll mess yourself up. So this equation helps you identify kind of ranges you need to go, and then in the design part, you bring that into play. And this is just a slide that actually shows the toughest thing with a topical agent. Basically, as I said, you really would like to have lowish molecular weight, high solubility, and moderate to high log P. Well, the two plots kind of show the problem. If you want low molecular weight, you're going to be in a very low log P range, low molecular area weight in log P for low log P correlate. But solubility doesn't correlate with that. So if you have a low log P, your solubility is going to be high, but you don't want a low log P for a topical agent. You want a moderate to high, and that's the problem. It's very hard to make a low molecular weight, lipophilic, highly soluble molecule for topical delivery. And that's why repurposing an oral compound can work, but if it's a molecular weight of 450 to 500, you're starting to have problems. If its solubility is less than 1 micromolar, you start to have real problems. So these are some of the things that you have to work around. Now, admittedly, you can formulate a lot of things, and you can work towards formulating those compounds, but it's more difficult. So tofacitinib and reluxantib have been evaluated topically, and they're not bad, because they're actually fairly good starting points, but many, many other kinase inhibitors and many other JAK inhibitors wouldn't be good. And yet you can still do better than ruxolitinib or tofa. So let's go back to the original tofa story. The box compound, this compound obviously Groton at the time didn't want to do anything with it because they weren't thinking of topical, they were thinking of oral for transplant. This compound is very lipophilic, very low molecular weight. Solubility is probably good because of its size, although I don't know it exactly. But this would probably be a point where you might, in a drug discovery program for a topical agent, start to diverge. And if you did that, you might end up with a compound very different from tofacitinib, but it might be a better topical agent. So that's what a medicinal chemist would think about, trying to do that. They take structure into account, and where would you go from there? And so that's kind of the process that we use. So basically, some of the messages I just want to leave you with, oral drugs and topical drugs have different properties. The steps you take to optimize for an oral compound don't necessarily help with a topical. In fact, you're often going away from the best space. You can fix the liabilities of oral drugs through formulation. We'll hear a little bit more about that in the next presentation. Metabolic stability is something you can design in if you want or not, and that really depends on your particular target. And basically, though, and it's happening more and more in the industry, the intentional design now of drugs for topical delivery should provide superior compounds for us.
All right. Thank you. Don't go anywhere. Don't go anywhere. Are there any questions for this? While they're lining up, one of the questions that I have is, should we be thinking about this in a different manner if, for example, you're trying regarding barrier function and designing compounds, if you're thinking about a disease like vitiligo in which you'd expect that the barrier function would be normal versus a disease like atopic dermatitis when you would consider it not to be normal? Any thoughts?
Yeah, exactly. When the skin is disrupted more, you can get away with a lot less of a permeable compound. So that's something we do take into account. You know, it really depends on what you're doing. And that ends up often requiring different formulations on top of it.
Exactly. Question?
Hello, James Clark from SimSit. You presented some ideas for predictive models such as Particle equation, which are fairly simple empirical models. Have you looked into using more advanced models such as physiologically based pharmacokinetic models? There's quite a lot available, both open source and from commercial platforms, which are considerably more accurate at predicting local concentrations.
We've done a mixture of that. We also do, you know, some basically in vivo assays using like a Franz chamber to do that. So we actually we kind of take the mathematical models and then we kind of go into a Franz chamber fairly quickly to kind of see the permeability there across pig skin or human skin, depending on the situation. But I'd like to hear about some of the other models you have in mind in case there's some I'm not aware of.
Yeah, I'd be interested to have a chat afterwards.
Sounds good.
Okay. All right, thank you very much. So once you've created your molecule, how do you actually formulate it in terms of topically? For that, our next speaker is Dr. Brezina Mikiak-Kohn, who's professor of pharmaceutics at Ernst Marios School of Pharmacy at Rutgers.
Well, good afternoon, everyone, and thank you to SID and Aclaris for inviting me to speak at this symposium. So what does a formulator think when they get one of these new molecules that we talked about? So first of all, in the broad spectrum of things, you have to think where your target is in the skin. So if we look at this first slide, we see two extreme ends to start with. First of all, there are preparations meant to stay on top of the skin. A good example that I think we're all using, particularly in the summer in large areas of the body, are insect repellents. And we hope that that preparation keeps that agent only on the top of the skin and repels the mosquitoes. But of course, actually in fact, those compounds usually get very well absorbed into the skin because of the chemistry that was just presented in our previous lecture. On the other hand, you may be designing a transdermal patch, and that, of course, is meant to use the skin as a route of entry into the body and basically deliver the agent across the entire layers of the skin and into the systemic circulation. And then you've got that gray area in between that we know as skin, epidermis and dermis, and we may want to apply anti-inflammatories, etc., to the various layers of the skin, and we hope that we can reasonably target agents, at least to the right layer where we need to exert the therapeutic action. So in this slide, what we're seeing is we've applied something to the skin. This particular example looks at something that has been suspended in a vehicle, and we'll discuss vehicles in a few minutes. The first thing we note that anything that is a powder is in suspension has to dissolve in the vehicle before any drug can get absorbed. And after that, of course, it has to, what we say, partition as formulators into the stratum corneum, and we'll talk about partitioning in a few minutes as well. And of course, we heard that a lot of the drugs these days are fairly lipophilic, and the stratum corneum has both lipophilic character and hydrophilic. So the lipophilic portions of the stratum corneum will tend to bind certain drugs within it and prevent further movement of those molecules further down the skin. Others, of course, pass through the stratum corneum fairly quickly, and of course that's a relative term, and we'll look at that in a minute too, and pass into the viable epidermis and finally into the dermis, and there they hit the microcirculation and possible systemic absorption, and of course metabolism, and also the possibility of binding. And I think we all know that steroids are the common example used of possible side effects of atrophy in the skin because of the binding that's inherent to those molecules. So we've decided on the target where we want to get our molecule to. What options do we have in dermatology? Well, a lot of what we currently have is really old. So we have liquids, we have semi-solids, we have solids. Emulsions, well, they've been known for a long, long time, but at least they allow us to play around with the idea of things dissolving in an oily environment versus an aqueous environment. And we have oil in water and water in oil emulsions. We have suspensions where things are suspended and then dissolve and penetrate the skin. We've got sprays that we can neatly apply the drug to the skin. Then you have to think of, what the packaging is going to be, the pump, the propellant, and of course we have solutions from which you hope that the drug is delivered. Then the semi-solids, those are the more elegant preparations from elegant creams that are oil in water or can be water in oil, emulsions, ointments have been around forever, pastes, foams and gels, aqueous and non-aqueous, and then of course we have the solids, the patches, the powders, again very old dosage forms, and sprays that actually deliver solid particles into the skin, and there's a lot of research in that area in the literature. So formulation facts, generally most vehicles are going to be blended from main classes. We've got aqueous, we've got a powder and oils. We've already discovered that in the last slide. And then we add things to it, and that's why we've got those long lists on all of the preparations. So we're going to be adding viscosity increasing agents, otherwise known commonly as thickening agents, emulsifying agents to stabilize those emulsions, buffers to make sure that the pH is fine, antioxidants, preservatives, perhaps propellants, etc. And among that whole process, we will be testing and optimizing the topical formulation. And the things that we have to think about from a physicochemical standpoint is some of the things that were already mentioned in the first presentations, the stability of the active ingredients. The stability, though, also of the excipients and any chemical interactions in that formulation that you're designing. Do you want to increase the viscosity of your preparation, or do you just want something that may be runny and watery, but it may not matter to that patient or to the condition that it's treating? Does it matter that you're going to lose water when things are applied or other volatile components? Are you going to have phase changes when you apply that formulation? Does it matter what particle size you have? Yes? Will it be better to nano size perhaps to increase what we call dissolution, or the process of the drug going into solution, and then the penetration? Do you need to change the pH? You may be working with a drug that ionizes. You have to make sure that obviously your preparation is well preserved and you don't want any particulate contamination that you don't desire in your formula. And of course other things play a role as well. How does the product look? Because you as physicians, and particularly the patients, do respond. There's a lot of psychology in what you put on your skin and where you put it and how it might work. Is there any odor? There may be a very nice, elegant preparation, but there's a smell to it that your patients aren't going to like. How easy is it going to be to remove from the container and apply? How well does it spread? When it spreads, how does it behave on the skin? And then does it produce anything at the end from any local irritation at the site? Does it leave the skin bright and shiny or oily and unpleasant? Ointments are an example. Patients don't like using them because they're greasy and they come off on clothing and all the rest that goes with that. So we already heard from the first lecture what a good drug candidate might be. And what we heard was molecular weights were mentioned. And of course, depending on what text you read, a lot of the books will say cut off at 500. Some are now saying a little bit more. Again, that middle range, if it's too high, you have to use other things than passive transport. So molecular weight around 300, 600 is fine, but there are exceptions, by the way. Again, I don't have time to go into this, but hyaluronic acid is 500K. Aptamers are 300K, and yet they go in passively into the skin in appropriate formulations. We also heard that we need good lipophilicity and suitable aqueous solubility. And log partitioning, usually we look at octanol water or other organic solvents, 1 to 3.5. Again, somewhere in the middle is good. If you go outside of those ranges, you have problems. And of course, we need to realize that any of the oral doses of any drug are not going to translate directly to the skin. Actually, it's very good, because in the skin you will need far less, because, for example, the first pass effect, the liver effects, don't take place. So you can really go down with the dose, and also if any of that drug gets absorbed, you're looking at less side effects, which is a good thing. What about the vehicle that then this drug is going to go into? We have a rationale in physical-chemical approaches how you want to think about successful formulations. So you need to maximize skin absorption. That's fairly obvious. Get past the stratum corneum barrier, but also solubilize the active in the vehicle. And solubilization is basically a formulator's, chemist's name for saying insoluble drugs can be made soluble if you know what you're doing. And the process is called solubilization. And the other process that plays a role in this is partitioning, and we'll look at that in a few minutes in more detail. But here it basically describes a ratio of solubility between the stratum corneum and the solubility in the vehicle. In simple terms, again, I've got an equation which is relatively simple. I avoided long equations. There are books on equations on this stuff, by the way, and on flux. The best way to look at it is to say, how much does your drug want to stay in the formulation versus leaving it and going into the skin? Partitioning is that process. Does that molecule want to leave this apartment and go to that apartment? And the more attraction it has, the better it will enter the skin. And of course, we know that drug concentration must matter. The driving force of getting a drug into the skin, of course, is its soluble concentration. You can also prolong your therapeutic effects in the skin by having a reservoir of the active trapped in, for example, a suspension. You know, there are 5-fluorouracil, for topical examples, where the preparation has a lot of insoluble powder, but that insoluble powder basically dissolves later and replenishes the drug that passes and gets transported through the skin. But each time we need to remember it's not the solid, it's the soluble portion that will be going through the membrane. Role of excipients. It's the majority of the stuff that goes into the formulation. So what is the role of those, what are known as excipients? They improve usually the solubility of your drug. They control drug release and permeation. They improve the general aesthetics of your dermal formulation and hopefully increase patient compliance at the same time. They improve the skin permeability and skin deposition and the stability of the formulation, and of course hopefully with preservation provide low microbial growth or no microbial growth and minimal contamination. And if you're wondering, what can I add to my formulation? Well, there's a place to start. You can look at the FDA Inactive Ingredients Database that provides a list of formulation excipients that's already present in formulations that are marketed and that patients use. It also gives you the maximum level of the product that's out there. And it's a valuable resource if you're looking for examples whether something may be acceptable if you're thinking of FDA approvals. There's something that a lot of formulators are now thinking about more, and probably in general the chemistry still hasn't really covered in great detail, is the fact that the formulation in the tube, in the jar, whatever the patient has in their hand, changes by the time they have put it on their skin. First of all, formulation components are being depleted. Your drug is being absorbed, hopefully, from that formulation, and the concentration that you thought was there originally isn't the same anymore. Things may be evaporating. You know, ethanol may be evaporating. Other components within the formulation are, in fact, or maybe being absorbed by the skin at the same time as the drug is. It may lead to saturation of your drug that you may not have predicted initially. Hence, the overall study here that I'm describing is looking at the microstructure of the formulation when it's actually on the skin at 32 degrees that skin is, and being used and possibly even being massaged in by use, so the application of force, which we don't often express it that way, but of course, if you're looking at the rheology of formulations, that's exactly what it is. And the microstructure of the formulation will change. So I kept promising that we'll go back to partitioning. So here's a look at partitioning coefficients again. So we've decided that most drugs, if you look at partitioning, do not penetrate the skin very well. And if they do, it's small quantities, and we may want to have more because we're going to lose some. Exfoliation, sweating, the patient obviously washes the area, may get absorbed onto clothing, etc., may even be degraded by the sun. So you can improve this partitioning by increasing skin hydration by some of those added excipients to the formulation, and classic examples include ethanol, propylene glycol, and various vehicles and agents that are called chemical penetration enhancers, or often correctly named penetration modifiers. So what happens with these partitioning stories? And I'm going to simplify what I have on this slide in very, hopefully easy terms. So on the left-hand side, what you see is the vehicle compartment. So let's think of that as one area or one particular compartment of the formulation. And on the right-hand side, we have the stratum corneum lipid, or the skin, that we're trying to get the molecule into. We've got a vehicle molecule, that's the open circle, and then the black molecule, which is your drug. So there will be a force. It's weak, but the vehicle and the drug will have some attraction for each other. But really what you would like is to have that black molecule end up in the stratum corneum and get absorbed. So you want to have what is known as the push effect, that molecule has been pushed into the skin and that pushing level is high. The other effect is listed below, and these are famous effects, you know, for physical chemical formulators, and very well known in the art. How you can manipulate these is to have the drug being pulled into the skin through attractive forces towards the stratum corneum lipids. And that's how partitioning basically is broken down into these two stages. And remember, the idea higher level is, does that molecule want to stay in the vehicle, or does it want to go in the skin? Because the real conclusion could be that I could prepare you a wonderful, elegant, well-preserved cream with a high concentration of drug that will never leave that formulation and pass into the skin. And I can prepare a very low concentration of a cream where most of the drug will want to go into the skin. And that's the art of formulation. But of course, we still sometimes get, by the time we do the Franz cells that were mentioned, testing, even clinical testing, you may have just poor inherent permeability, and you may want to think about adding some additional agents called these chemical penetration enhancers, or even form carrier systems. You may get poor or no drug release from the formulation, so you've made that elegant cream, but nothing is actually released into the skin. Or you may have a very low dose, entering the skin, and you may still have an option of increasing the concentration in the formulation. And you may have drug metabolism that you may or may not be able to control, for example, by making a prodrug. So what are chemical penetration modifiers? There are books on them. There are various classes of agents. You may be aware DMSO, dimethyl sulfoxide, was one of the old ones. And there are agents that you use every day and your patients use every day, like ethanol in formulations, that it itself is a penetration enhancer, even though it's a very well-known vehicle. Likewise, transcutol, that you probably are all familiar with. But there are many available, many in the literature and some in marketed formulations. So let's now narrow things down and say, what's a good drug candidate? What about the JAK inhibitors? We were talking about the fact that we need good lipophilicity, suitable aqueous solubility, and a certain molecular weight. Well, as my previous presenter mentioned, a lot of JAK inhibitors do have a reasonable molecular weight. They're not like in the thousands, so you have to think of some other ways of delivering to the skin. Log partitioning in, depending on which molecule you look at, may be good. And again, a good permeant, you have to have reasonable flux, in other words, transport into the membrane. And again, we've already mentioned that the oral dose doesn't give you an indication of what you need in the topical. So let's finish by looking what's happening right now with a few highlights. So if we look again, this is from 2017, we have a systematic review of what the papers have published at that time, and we start seeing things about JAK inhibitors and psoriasis, alopecia areata, and that they have the potential to be used in dermatology. We see papers like this from, I think this is 2018, that say mostly JAK inhibitors are used orally. However, there are articles and potential research projects that are leading to their being used in dermatological conditions, but using the skin as the port of entry. Here, I think this is a 2018 paper. We see that studies are starting up in humans, and we look down at 6 patients. It's a small study, and an ointment was being used and compared. Again, ointment, old preparation. Perhaps, if successful, we could move away from ointment because, again, it delivers well, occludes the skin well, and that was probably what this clinical trial wanted to do, make sure that the agent went in, but there may be a more elegant way of packaging that active agent and yet still deliver adequate amounts to the patient. And we also see papers like this. This is 2017, where people are even putting things in all kinds of wonderful carriers, and I could pull up a lot of more papers in this area. In other words, packaging it, wrapping up the drug, in other words, and hoping that that carrier will then deliver it and target it to the right area of the skin, and also make the molecule more stable. And finally, to finish off, since this is an Aclaris-sponsored session, I thought that looking at the website, we have already things that are looking very successful in phase 1, 2, and 3 of the JAK inhibitors. So I'm looking forward to hearing a little bit more of what new is coming along in the rest of the session. And with that, I thank you all. And just a picture of my group. Obviously, they contribute to all of the other things that we do in my lab that's involved with formulations, optimization, measurement. We also do skin engineering, tissue models. But I thank them for being there and supporting all the stuff that I do, both in the lab with nearly 20 people, actually a lot of people are missing here, and the Center for Dermal Research, where I do events, symposia. There's one coming up, and workshops that we're doing with the FDA in June. So I hope to see some of you there. And with that, thank you very much for your attention.
All right. Any questions from the group? While we're waiting, I actually had a question. So it looks like a lot of what we're seeing in formulations, a lot of it is old and tried and true, propylene glycol, things like ethanol. What do you see the big areas of growth in this area? Like, for example, when Aclaris sponsors the 10th annual symposium, what will we be talking about?
I think different formulations with different carriers, and the work is already being done. I think it's the kind of balance between, you know, what companies I think believe they can pass through the FDA and that's acceptable, that patients have used, that you know, that's a safe thing in quotes, versus going out, you know, where the research is, possibly looking, you know, like these gold particles,
right,
and trying to actually get that to market. That's always been this big balance of, you know, do we do the old thing that we know works and that the FDA won't question and we can get approved, versus can we get this new agent in this new formulation, and by the way, possibly a new device. I mean, that's where we're going with a lot of the research. I mean, I think everybody's probably goes to other symposia and meetings. You know, the microneedles are of interest to deliver things because they poke the skin in an elegant way. You can apply all kinds of new formulations. We hear about all these nanocarriers that are very successful, and there's enough proof already in the literature, but now there is the question that, you know, which company is going to take the risk and try and get this through the FDA and through clinical trials that are so expensive, and get the investors to back it up too. So we'll see, but I hope we'll have a lot of new things coming up.
Good. All right. Well, thank you so much for both of the speakers.
Thank you very much.
So I'll ask Dr. Brian Kim to come up and moderate the second part, which is the more translational aspects.
Okay, good afternoon. Welcome to the Aclaris Symposium. Thanks to Aclaris again for continuing to sponsor this session. It's always great to hear about new applications for inhibiting this very exciting pathway. Just a brief comment. Although JAK inhibitors really didn't start in dermatology, I do think, based on the prior speakers, that this class of drugs has the potential to revolutionize the field of dermatology more profoundly than potentially any other specialty of medicine. So I think it's very exciting. And one of the things that I've really enjoyed about learning about JAK-STAT signaling pathway is that we're now finding there are so many other applications and fundamentally changing how we view the biology of this pathway. And that will be highlighted here today in this session. We'll be hearing about JAK signaling in the context of infection, fibrosis, as well as cancer. And I'd like to also just highlight that the next talks will be very brief, about 7 minutes per speaker. So I ask the speakers to be on time, please. And then we'll start with Matthew Brewer from Rochester, who will be talking about JAK kinase inhibition in the context of a vaccinovirus infection.
So thank you everyone for coming to my talk, and thank you SID and Aclaris Therapeutics for giving me the opportunity to share my research with you guys. So today I'm going to take a completely different avenue on what we can actually use JAK inhibitors for, and what we're interested in is how they can prevent infection in the context of AD in individuals who see viral infections within the skin. And so the only conflict of interest that I need to disclose is our group has filed a provisional patent application looking at ways to minimize epithelial infection. And so to begin this talk, I really have to orient you to some of the characteristics of atopic dermatitis that have driven this research. So we know from clinical studies that these individuals experience barrier disruption within the skin, both at the level of the stratum corneum and within the stratum granulosum in the tight junctions that are formed. Additionally, these individuals have elevated levels of Th2 cytokines. Important for this talk today is IL-4 and IL-13. And when Th2 cytokines and barrier disruption are taken together, we know this can possibly lead to pathogen invasion. So of interest to our group is vaccinia virus, and we're interested in this because we know that individuals with atopic dermatitis can develop this complication known as eczema vaccinatum when they're exposed to the smallpox vaccine, which contains vaccinia virus. And to put this in context today, as of January 2019, we know there's about 330 million people in the US. Of this, there's roughly 30% of patients, both 20% adolescents, 9% of adults that have AD. And so when you break that down, there's roughly 33 million people that have AD. And so the predicted occurrence of eczema vaccinatum is about 1 in 100 people, and really importantly, the fatality rate is 30%. So in the event of a clandestine release of smallpox, we're looking at somewhere upwards of 100,000 deaths from people who may be exposed to that vaccine that are in the AD population. And so what we know from biology and JAK signaling is it's really important in the downstream effects of IL-4 and IL-13. And so for the sake of this talk, I'm going to really focus on this one molecule called pyridone 6. So it's a pan-JAK inhibitor that we know should block all JAK molecules in this pathway. And so first, to develop a system where we can test whether JAK inhibitors are important in vaccinia virus infection, we really have to understand our model. So we in the Beck lab use primary human forest skin keratinocytes as our model for skin. And what I'm showing you in this data pick is really that when you differentiate these cells, as is shown in the black line on the left, they form barrier, which we can measure by transepithelial electrical resistance. The red line really shows you that when you expose these same cells to IL-4 and 13, you have a delay in barrier formation. And to give you a little bit of an idea of the variability we see within donors, on the right I show you for every line that is an individual primary keratinocyte donor. And the majority of the people that we use in these assays really respond by decreased here. So taking this a step forward, what we were really interested in is when you expose keratinocytes to IL-4 and 13, do you see increased infection with vaccinia virus? Essentially, can we model eczema vaccinatum? And so on the left, what you'll notice is when we just differentiate keratinocytes and media alone, you see little pockets of cells that become infected, and that's highlighted by the green fluorescence. On the right, if we add IL-4 and 13 into this mix, you see this largely disseminated infection. And really what I'm showing is the red and the blue highlight the fact that vaccinia gets in, it fully replicates, and you release new virus, which does not happen in a normal context. When we look at a variety of different donors, you see that we have significant increase in plaque number after addition of IL-4 and 13, showing that this is a relatively generalizable phenomenon in our system. So now trying to incorporate JAK inhibition into our system, we first wanted to understand can we reverse this loss in barrier formation when we add JAK inhibitors. So the image that I'm showing you right now is keratinocytes exposed to IL-4 and 13. 48 hours later, we add a JAK inhibitor, and 24 hours that we measured here to look at barrier function. And so you can see that after incorporation of 2 different concentrations of a JAK inhibitor, we really are able to restore barrier formation, reversing this loss that I have shown you previously. Importantly, when you remove the JAK inhibitor, levels fall right back down to IL-4 and 13 treated controls. So you really need this sustained inhibition to maintain this phenotype. And so finally, the most provocative data that we're currently working with is the fact that using our infection model, can JAK inhibitors really reverse the susceptibility to vaccinia virus? And so if you look across this and you really focus on the blue bar, what we're showing here is cells that have been treated with IL-4 and 13 and then exposed to a JAK inhibitor right before they're infected. And so as we add an increased dose of our pyridone 6 or our pan-JAK inhibitor, you can see that we really diminish viral infection quite substantially. And so just to highlight that, you can see representative plaques on the bottom really showing the change from just the controls down to the JAK inhibitors. And so in summary, I hope what you've taken away from this is that Th2 cytokines really are able to increase keratinocyte susceptibility to vaccinia virus, that JAK inhibitors can reverse the loss in barrier after Th2 cytokine treatment, and that we can possibly use JAK inhibitors to minimize viral infection in skin cells. And so the next steps in our lab are really to look at what JAK molecules are really important in this phenotype. So there are many FDA-approved and late-stage clinical trial JAK molecules that we're hoping to incorporate into the system and better figure out what target would be the best one to hit. And so I just want to acknowledge my PIs, Dr. Lisa Beck and Dr. Ben Miller, our collaborator, Dr. Brian Ward, he's the pox virus expert, and then funding from U of R. And if anyone's interested in this, please come see my poster, 479. I'm happy to take any questions. Thank you.
Thank you very much. If there are any questions, please come up to the microphone. One question is this, do you think this is relatively specific to certain viruses, or is this a broader antiviral phenomenon?
So what we're hoping is eczema vaccinatum is a historical aspect with AD, but eczema herpeticum and eczema coxsackium are more of the pathologies you see in the clinic today. So we have access to herpes virus, and we want to repeat all these studies with that to see if it's more of a broad phenotype across a variety of different viruses.
Another question, please.
Thank you. Yves Poumet from Belgium. If I understand correctly, you tested primary cells in monolayers.
Yes.
Did you try to put them into a reconstructed tissue in order to get insight whether the receptor will be differently expressed in those conditions?
Yeah, so we have only used monolayers. We have access to people at U of R that can actually make reconstituted skin substitutes, but at least for proof of concept studies, we wanted to start with this because that way we get rid of the stratum corneum, which we already know is disturbed.
The sensibility to IL-4, IL-13 is enough in monolayers?
Yep.
Thank you.
Yeah, no problem.
Okay, one more question in the back.
Dominique Pichard from Bethesda. Question, we know that JAK inhibitors, when taken systemically, one of the biggest risks is viral reactivation. So why is it that you think that in the skin it can be protective?
So going off essentially what we just heard, right, we can reformulate these as topical applications. So we're hoping to circumvent some of those issues. And also one of the things that I couldn't really highlight here is we really just think this event is specific to keratinocytes. And so we don't even want to hit other cell types. So if we could change the chemistry of JAK inhibitors to just get them to sit in the epidermis, then we think this might be a really useful therapeutic for people that have AD and experience these infections.
Thank you very much. Our next speaker is Joanna Jackow from New York. She'll be talking about ruxolitinib in the context of tumor growth in recessive dystrophic epidermolysis bullosa squamous cell carcinoma.
Hello. Thank you. I would like to thank the organizers for inviting us and giving the opportunity to present our preliminary data on dystrophic epidermolysis bullosa and using the JAK inhibitor. Epidermolysis bullosa has a long history in our laboratory, and this is inherited skin disease that is characterized, as you can see, with blister and scarring, and can be inherited recessively and dominantly. And I have to point out that patients with the recessive form that are at great risk of developing squamous cell carcinoma, as you can see on the clinical pictures. And all forms of dystrophic epidermolysis bullosa are caused by mutation in type 7 collagen, which is a large gene encoding large protein that is secreted by keratinocyte and fibroblast in the skin. And this molecule undergoes proteolytic processing to form homotrimers that beat anchoring fibrils, as you can see here on the electron microscopy picture with the red arrow. And patients that have mutation in type 7 collagen, with a null mutation, this leads to complete absence on anchoring fibrils, and this leads to blister formation in the skin. Now, the blister formation actually occurs constantly, and this leads to non-healing wounds in the skin of these patients. And this impairs keratinocyte migration and fibroblast migration in the skin lesion. And this defective wound healing leads to inflammation, bacterial infection, and increased TGF-beta-1 in the skin, which then affect conversion of fibroblast to myofibroblast. And this process that has been described in several papers leads to upregulation of JAK-STAT signaling. And now this hasn't been tested yet in EB patients, and our expertise in JAK, especially ruxolitinib JAK1/2 inhibitor in the laboratory, lead us to propose this hypothesis if we can use JAK1/2 inhibitor and prevent tumor growth in this RDB-SCC tumor. So in the JAK-STAT signaling cascade, when we used ruxolitinib, we blocked, as the previous speaker showed, we blocked JAK1 and 2, which is here in this step, in the signaling pathway. For doing this experiment, I would like to acknowledge Dr. Andrew South, who kindly provided us with published in very well characterized RDB-SCC cell, spontaneous immortalized, that we actually used for these studies. And first of all, we demonstrated that this signaling, JAK-STAT signaling, is upregulated in our internal SCC39 cell line, as well as in those used RDB-SCC cell line. As you can see here, phosphostat and is expressed compared to the normal human keratinocytes where there is no signal. Then, first of all, we used the individual selective JAK1 and JAK2 inhibitors and demonstrated we can block STAT3 phosphorylation. However, there's no effect on proliferation. When you can see here, there's a normal signal on cyclin D1 expression. Then we used the ruxolitinib, the JAK1/2 inhibitor, and indeed we could reduce the cyclin D1 expression and would also decrease the STAT3 phosphorylation in the SCC39 line. Also here, the inhibition of JAK-STAT signaling using ruxolitinib, we could demonstrate in vitro that the proliferation is dependent on this signaling, and over the period of 48 hours, you can see here the proliferation is diminished in those cells. And finally, we recently used the RDB-SCC tool line and used also ruxolitinib to target JAK1 pathway and demonstrate that phosphostat is decreased in those cells. And now we wanted to go in vivo and demonstrate this in reality in the skin, how we can use this application. And meanwhile, we developed a new model which we called preclinical xenograph tumor model for RDB. And we used our expertise in generating skin constructs, and you can see here we used fibroblasts from EB patients and RDB-SCC cells that I showed you before, and produced this beautiful skin construct within 14 days in the cell culture and subsequently grafted on mute mice. And you can appreciate it here that at day 25, these tumors look really like resembling the human RDB-SCC tumor in patient. So we further characterize these tumors, and we could see invading columns of the tumor into the dermis, and also in the panel B you can see in upper dermis there is differentiated tumor cells. And also immunofluorescent staining confirmed the expression of phosphostat in the RDB-SCC graft. There's absence of collagen 7 expression, proving that these cells are coming from RDB patients. And we also demonstrate the expression of epidermal markers such as keratin 14, 10, E-catarine, and proliferation markers, P63 and K67, which are ideal readout also for screening after the drug application. So, moreover, we've been successfully transplanting this tumor on new nude mice, giving us the opportunity of developing a platform for drug screening. And so we recently started the first experiment in which we applied ruxolitinib orally via oral gavage in our mice. And the treatment the dose was 70 milligram per kilogram once a day for consecutive 4 weeks. And we've been having a small cohort of mice, but the overall conclusion is that we could measure tumor reduction over this period of time, or some of the tumors just stop to grow. This is a picture of representative images taken from the mice, and also by calculating the tumor volume, we significantly can see the reduction in the cohort of mice that were treated with Ruxo. And this is a Western blot from the harvested tumor tissue and tumors that were treated with PBS, demonstrating that we're targeting the phospho-STAT3 signaling. So in the conclusion, I hope I show you here therapeutic benefit for RDBSCC using the oral administration of JAK1/2 inhibitor. And over this period of time, when we treat the mice, we did not observe any toxic effects on the mice. And we think that it's a promising new anti-cancer drug that can be used for RDB patients, but we can also go and use in another cancer settings. And with that, I would like to thank you, my mentor, Angela Cristiano, and the laboratory and our EB Research Foundation Consortium that we work also on EB with another project, and thank you for your attention.
Thank you very much. Any questions from the audience? Yes.
Hi, Joanna. That was a beautiful talk. Some really nice data you have there, and certainly fits with our understanding of JAK-STAT signaling and the progression of squamous cell carcinoma. I offer a word of caution from translating the data into patients. I've had discussions with Alex Nystrom in Freiburg, who's used ruxolitinib to treat the hypomorphic mouse model of recessive dystrophic EB, and he reports huge toxicity in that animal model. The drug exacerbates the wounds and leads to earlier mortality. So I would urge you to reach out and speak to Alex about his experiments. He's not published that data, so please speak to him before considering taking that to the patients.
Thank you very much for this comment.
Thank you. Another question in the back.
Yeah. Thank you for that great talk. I have two questions. One is that did you take a histology at the end of your treatment? And the second question is that have you observed the mice after you discontinue the treatment? Does the tumor grow back?
Yes, I have taken histology. This is in processing. And after this treatment stopped not long time ago, so I'm still observing every week and taking pictures. And I've been dissecting some of the mice to transplant to another mice to continue the treatment.
Thank you.
Thank you.
Okay. Thank you very much. Our next speaker will be Yuhan David Gu. He'll be talking about targeting JAK-STAT pathway in the context of cutaneous T-cell lymphoma. Oh, did we switch the order here?
Yeah, thank you, Brian. And I would like to thank the organizers for giving me the opportunity to kind of present some of our data. So I'm going to show you some of the data that we have been generating from a disease called lichen planus. So lichen planus, it's not an uncommon disease. It's actually found like in 0.5 to 1% of the population. It tends to be a chronic immune-mediated skin disease of unknown etiology. As you can see from the photographs on the right side, it's characterized by polygonal, flat-topped, violaceous papules and plaques. And they have this overlying reticulated fine white scale, which we call Wickham's 3A. It can affect any portion of the skin. It can be the skin, nails, hair, and even the mucous membranes. This disease tends to have a major impact on quality of life, particularly in those patients that have oral erosive lichen planus. And with chronicity, it can increase the risk of malignancy. So what we did is we have access to paraffin-embedded tissue. So we found, we obtained 24 healthy control skin samples, 20 classic lichen planus samples, and then 17 hypertrophic lichen planus. And hypertrophic lichen planus is the hyperkeratotic form of lichen planus. We did a microarray profiling, and you can see that on that principal component in the left upper corner. And you can see that the normal skin shown in blue separates from both classic and hypertrophic lichen planus. And lichen planus and hypertrophic lichen planus overlap, as you can see on the right. but the overlap between the two in terms of gene expression was highly correlative with a correlation value of 0.85. When we looked at the biological categories that were enriched in this data set, we could see that the most significantly enriched pathway was interferon gamma-mediated signaling pathway, and that was both for lichen planus and hypertrophic lichen planus. We also see increased categories such as defense response to virus, T-cell co-stimulation, adaptive immune response, apoptotic process, and then keratinization. And the keratinization was more highly enriched in the hypertrophic lichen planus, as you would expect for a highly keratotic disease. We looked at the expression of specific cytokines. We focused on type 2 interferons, interferon gamma, and that was significantly increased in classic lichen planus and close to being significant for the hypertrophic LP. And both receptors for interferon gamma were markedly increased. Same for the interferon response gene. So we looked at MX1, MX2, and OAS1 and OAS ligand, and these were significantly enriched in lichen planus skin. And then we looked at the downstream responses. Could we see enrichment for responses downstream of the type 1 and type 2 interferons? And as you can see from this graph, you could get the normal skin right in the lower corner, and then you get the disease samples kind of going up like a comet tail. So we get both type 1 and type 2 interferon enrichment in these lesions. But if you look at IL-17 responses and IL-4, it's much more muddled kind of a picture where you see much less clear pattern. If you look at the expression of the type 1 interferons and the receptors, interferon receptor 1 and receptor 2, you can see that there certainly are changes going on, but nothing that was significant. We also looked at the expression of type 3 interferons. And again, not much happening with that. And in terms of staining, we did staining for the type 1 interferons, interferon alpha, interferon beta, and interferon kappa. And not much going on. There was a slight increase in interferon kappa expression in the epidermis, but not nearly as prominent as what we have seen in other inflammatory skin diseases such as lupus. And if we look at the transcriptomic network, so one way to kind of analyze data like this is to kind of take the complex gene expression pattern and kind of like establish a network to kind of find the nodes that are kind of the key drivers of the pathogenesis. And when we looked at the gene expression pattern, we can actually see these nodes that you can see on this graph. So you can see how the gene expression and the genes kind of center around interferon gamma, JAK2, STAT1, and even STAT3. If you look at the expression of the STATs in lichen planus, you can see that STAT1, STAT2, STAT3, and even STAT4 are increased in lichen planus skin. And if you look at immunohistochemistry and kind of focusing here on STAT1, you can see that STAT1 is certainly increased both in the epidermis and the inflammatory infiltrate of lichen planus. But if you look at phospho-STAT1, which is the measure of like an activated STAT1, most of it actually is happening right at the junction between the epidermis and the dermis, and that's where most of that inflammatory activity actually is going on in this disease process. To kind of study this in greater detail, we used a model system where we grow keratinocytes from one individual, and then we add in PPMCs from a different individual. It's similar to the mixed lymphocyte reaction where you have the cells kind of interacting or acting against each other, but in this system it's kind of one way. The PPMCs should be attacking the keratinocytes. So what we did is we used the system and then we added in the PPMCs, and as you can see from the graph, you see the flow diagram and then the quantification of that. If we pre-stimulated the keratinocytes with interferon gamma, we didn't see much increase in cell death. And same thing, if we just added PPMCs, we didn't see much of an increase in apoptosis, but if we primed the keratinocytes with interferon gamma and then added the PPMCs in afterwards, we could see marked increase in cell death in our model. Interestingly, even though it's not shown here, if we primed the PPMCs with interferon gamma and not the keratinocytes, no increase in cell death was seen. You can see the same thing. This is a tunnel staining, which is a measure of cell death in our culture system. So if you see keratinocytes alone, you see practically no cell death. But if you add in interferon gamma, you see much fewer cells, and you begin to see these red dots that are indicative of cell death. Interestingly, if we we could only get that induced or increased cell death by priming with interferon gamma along with the PPMC. So if we did type 1 interferons like such as interferon alpha or interferon beta, we did not see the same increase in cell death with the PPMCs. So suggesting that this process is specific to interferon gamma. We think about cytotoxic responses being usually MHC1 or MHC class 2 dependent. So we looked at the expression of the three HLA type 1s, HLA-A, B, and C, and then 3 of the MHC class 2 molecules, HLA-DR, DQ, and DP. And all of these were markedly increased in lichen plantar skin, and you can see the immunofluorescence on the right. We see increased expression of MHC class 1 in the epidermis and MHC class 2 on the inflammatory infiltrate. Interferon gamma is a very strong and effective inducer of both MHC class 1, particularly on keratinocytes, and it also induces a little bit of HLA-DR on keratinocytes. So what we did is we used the same system with the PPMCs and the prime keratinocytes, and then we started to kind of block the different pathways. So if we block MHC class 1, we could practically completely eliminate the cell death in our system. If we blocked MHC class 2, we got some decrease, but much, much less than blocking class 1. And same thing if you looked at the cell types that are kind of the driver of the disease. We blocked CD4s, NK cells, and then also CD8s, and it was only the blockage of CD8 that actually prevented cell death in our model system. And this is, CD8 T cells are actually very prominent in lichen planus. I don't have the data to show you here, but so this fits well with what we see in lichen planus. So one thing that we did, as I showed you before from the gene expression modules that we found in the network, some of the key nodes in the network were actually JAK1 and JAK2. So we generated keratinocytes using a CRISPR-Cas9 knockout, and we deleted JAK2 and STAT1. We also did STAT2 and JAK1, but I'm not showing that data here. We primed the keratinocytes in the same way with interferon gamma, then added in the PPMCs. And what you can see is in the absence of JAK2 or STAT1, we see no cell death. So inhibiting or deleting these genes are protective against this immune response. We did a drug target enrichment analysis on our differentially expressed genes, and this is the drug targets that were significantly enriched. So chloroquine, kind of related to Plaquenil, is a major medication that we use to treat this disease, and that was significantly enriched as a potential target. Prednisolone or steroids, highly enriched, and again, a mainstay of treatment for cutaneous lichen planus. But the one that kind of came by far the highest was actually JAK inhibitors. And what we did is we used the model system again, and now we, with the priming of the keratinocytes with interferon gamma, and then added in the PPMCs, and then we used the JAK1, JAK2 inhibitor baricitinib. And as you can see from these data here, the one that are marked with the red box, baricitinib treatment of the system completely prevented the cell death. Just to summarize, so I think I have kind of shown you that lichen planus is a debilitating chronic inflammatory disease of the skin and mucous membranes of unclear etiology. Lichen planus has heightened type 2 interferon response, and transcriptomic activity in LP centers around the interferon gamma, JAK2, and STAT1 signaling. Interferon gamma promotes cytotoxicity and is dependent upon JAK2 and STAT1 signaling, and can be effectively inhibited by baricitinib, a JAK1, JAK2 inhibitor. And taken together, this work suggests that JAK inhibitors can be a promising therapeutic avenue for patients with lichen planus. So that concludes my talk. I want to acknowledge some of the key members that have helped to kind of drive this work forward, particularly Dr. Shao, who did most of this work, and then other members of my lab and colleagues. And then last but not least, I want to also acknowledge the funding support that I have had over the years. So thank you.
Thank you, Johan. Time for one quick question.
Nice talk, thank you very much. Due to the systemic JAK2 inhibition cytotoxicity, I guess the question I have is, do you see lichen planus as a systemic immune imbalance, or do you think it can be treated topically?
Yeah, great question. I think probably both. You know, many cases that I see with lichen planus have just limited skin involvement on the wrist and maybe on the trunk, and that would be perfect for like topical approach. But then other patients that I have seen have fairly extensive either vaginal or oral and mucosal disease, going all the way down to the esophagus. So I have quite a few patients that are on Cellcept and Plaquenil as a combination treatment. And even at that stage, they actually struggle. So I think for a selected subset of patients with LP, I think a systemic treatment would be warranted. But I think for a good proportion of patients, topical approach would be probably appropriate or more appropriate.
Thank you. One quick question, actually. Did you see in your transcripts any cytolytic molecules like enriched perforin, granzyme?
Yes, those are increased
very much.
Yeah, we do see that.
Thank you.
Okay, thank you.
Okay. Looks like our next speaker is actually Swati Bhattacharya, and she'll be talking about pharmacologic inhibition of JAK-STAT with tofacitinib in the context of fibrosis.
Thanks for giving me opportunity to talk about share our research on JAK inhibitor and JAK-STAT signaling in scleroderma. Fibrosis in scleroderma leads to failure of skin, lung, and other internal organs, accounting for substantial mortality. However, so far it has no viable, the problem is that there is so far no viable therapeutic strategy. IL-6 JAK-STAT signaling pathway is implicated in numerous inflammatory and autoimmune diseases, including scleroderma. However, there is a scleroderma preclinical trial with IL-6 targeted therapy showed modest and variable clinical benefit, which leaves a room that could be a downstream JAK inhibitor, tofacitinib, could approve which is already approved for many autoimmune disorders, could be a novel therapeutic approach for scleroderma as well as fibrosis. So the objective of our research are to measure JAK-STAT signaling in scleroderma patient biopsies and examine the effect of JAK inhibitor tofacitinib in preclinical models of organ fibrosis. Now first we measure JAK-STAT signaling in scleroderma patient. To do that, we derived JAK-STAT signature gene from Broad Institute Molecular Signature Database, which is a coherently expressed signature that derived for many molecular signature database that represents well-defined biological state. From there, we found 150 JAK-STAT signature gene, out of which 87 genes are present in our scleroderma, publicly available scleroderma skin biopsy transcriptome dataset. And here we showed that compared to healthy control, this inflammatory intrinsic subset showed a significantly enriched JAK-STAT signature gene. Next, we asked that whether JAK-STAT itself are activated or not. For this, we measured tyrosine phosphorylated JAK1, JAK2, JAK3 as STAT3 in scleroderma skin and then lung biopsies. So first we measure phospho-JAK1, JAK2, JAK3 and STAT3 in scleroderma skin biopsies. Compared to healthy control, we found there is a significantly elevated phospho-JAK1, 2, 3 and STAT3 in the dermis. However, epidermis shows similar level of expression. And here we showed quantitation of this analysis. We showed a significantly elevated JAK1, JAK2, JAK3 and STAT3. Next, we measured these phospho-JAK and STAT levels in lung biopsies from scleroderma patient with interstitial lung disease. And what we found that compared to non-SSC control, this SSC ILD patient showed significantly elevated phospho-JAK1, JAK2, JAK3 and STAT3. Here is the quantitative data. Here we use 7 SSC ILD patients and 4 non-SSC controls. So here we clearly showed that JAK-STAT pathway is activated in scleroderma. So next we decided to look into this, what is the effect of JAK inhibition in preclinical models of organ fibrosis. So to induce fibrosis, we injected mice with bleomycin by daily subcutaneous injection, followed by a week of recovery. And we injected tofa daily concurrently with bleomycin. And at the, day 22, we harvested skin and lung. So here is the result from skin section from these mice. Here you could see that compared to vehicle-treated control, the bleomycin, where we see a clear dermis as well as a white adipose tissues, bleomycin induced a huge increase in collagen accumulation as shown by tichrome stain, increase in dermal thickening, loss of adipose tissues. And tofacitinib inhibited dermal thickening, then less collagen accumulation, and less inflammatory cells as well. And here we confirm, here you could see that there's a significantly decreased dermal thickness and then less collagen accumulation, significantly less collagen accumulation compared to bleo. We then measure macrophage activation in the skin sections by F480 staining, and bleo-induced significant increase in macrophage accumulation, which is significantly inhibited by tofacitinib. We measured alpha smooth muscle actin activation, which is also myofibroblast activation marker. Bleo-induced alpha SMA accumulation is significantly prevented by tofa treatment. And here we showed the effectiveness of this drug by showing that bleo-induced phospho-STAT3 activation is totally prevented. We next measure that the effect of tofacitinib in bleo-induced lung fibrosis. Here you could see that compared to vehicle-treated control, where we see a very clear alveolar architecture, bleomycin induced a total disruption of this alveolar architecture and huge increase in collagen accumulation, as you could see from tichrome staining. And tofacitinib significantly prevented this collagen accumulation. As you could see the fibrosis score, there is a significantly decreased fibrosis score. And here we confirm that by measuring collagen gene expression analysis. Next, we check the alpha smooth muscle activation in this mouse skin. And what we found that bleo induced a huge accumulation of alpha smooth muscle actin in a more fibrotic area, and tofacitinib significantly prevented that. In less fibrotic area, we see increased number of macrophage and lymphocyte accumulation, which is significantly prevented by tofacitinib, and then we showed that effectiveness of the drug by measuring phospho-STAT3 level. So this bleo-induced model is, we treat as an inflammatory model of fibrosis. We wanted to check that what happened in non-inflammatory model of skin fibrosis. For this, we used a genetically induced model of fibrosis, which is called tight skin mouse model, where, which is caused by large inframed genomic duplication of fibrillin gene that causes a 3 kb longer base transcript level. And these mice develop a homozygous mice die, but heterozygous mice develop skin fibrosis. So we can see a phenotype that they develop tight skin in the interscapular region, so it is hard to hold the mice even on the back. And then they show the increase in hypodermal thickness. As you can see from the picture, and most importantly that these mice lack early inflammatory stage of the disease. So here you can see that there's hypodermis of this mice is thickened, and which is also associated with p-STAT3 activation. And when we treated this, we started injecting tofacitinib at 5 weeks of old age of mice, and then we continued until week 10, and then harvested skin. And you could see that this hypodermal thickness is significantly reduced in tofa-treated mice. And it is also associated with less phospho-STAT3 activation. Now to summarize, we could summarize that in gene set enrichment analysis indicates the enrichment of JAK-STAT signature in diffuse inflammatory subset of patient. And we showed that phospho-JAK and phospho-STAT3 levels are significantly elevated in scleroderma skin and lung biopsies. And JAK inhibitor tofacitinib attenuated bleomycin-induced, which is present in inflammatory stage of the disease, as well as a non-inflammatory skin fibrosis in TSK mice. In conclusion, so what we thought that maybe JAK-STAT signature in skin might represent a potential biomarker to identify inflammatory subset of patient, and tofacitinib treatment might represent a novel therapeutic strategy in that particular subset. And finally, I would like to thank the co-workers for this project and also the funding agencies.
Thank you very much. Any questions, please?
I have a question back here. Did you use any positive controls like a methotrexate, steroid, or mycophenolate for your animal model study?
No, this is the first experiment we did so far. I mean, we plan to do more experiments, but yeah, we don't have the data.
Hi, I have a question too. Did you look at STAT6 signaling in your model? And also, in terms of the macrophage infiltration, did you look at M2 macrophages?
No, we didn't do a lot of mechanistic study because we first wanted to see that whether targeting JAK-STAT could be therapeutic for fibrosis or not. But we plan to do more mechanistic studies though.
Thank you very much.
Okay, thanks.
Okay, and I think we don't have another speaker. Okay, Yuhan Gu will now talk about cutaneous T-cell lymphoma and JAK-STAT signaling.
Hi, good afternoon. My name is David Gu. I'm a research fellow and current fourth-year medical student. Over the past year, I've been working with doctors Larissa Gaskin and Tereso Palomero investigating the role of the JAK-STAT pathway as a potential therapeutic target in CTCL therapy. I have no disclosures. Cutaneous T-cell lymphomas, or CTCLs, they are a poorly understood group of lymphoid malignancies characterized commonly by skin infiltration of malignant CD4 positive T cells. While it's a rare disease, there's been a consistent increase in incidence since the early 1970s. The two most common subtypes of CTCL are mycosis fungoides and Sézary syndrome, which is a more aggressive leukemic variant, often with a very poor prognosis. And while there are various skin-directed and systemic therapies that have been FDA approved, they have shown limited efficacy and, importantly, none are curative. Romidepsin, which was recently approved in 2009, has been one of the most promising therapies for CTCL. It's a potent histone deacetylase inhibitors with pleiotropic effects, but the exact mechanism underlying its activity hasn't been fully elucidated. However, patients on romidepsin have a median response duration of approximately 14 months, and development of resistance is very common. And Dr. Gaskin and Palomero have been investigating the mechanisms of response and resistance to romidepsin, and RNA expression analysis of patient samples and cell lines that have been treated with romidepsin showed consistent downregulation of JAK-STAT-related signatures, which suggests that the JAK-STAT pathway may be mediating romidepsin's mechanism of activity, and that constitutive activation of the JAK-STAT pathway could lead to development of resistance. In addition, there have been recurrent mutations in signal transduction pathways, which have been identified in genomic studies. This is a mutational analysis of 137 CTCL patients across 7 sequencing studies. It highlights the heterogeneity of the disease with recurrent copy number alterations, insertions, deletions, and other coding mutations and key oncogenic pathways. Importantly, I want to highlight that copy number gains were found in STAT5B and other mutations in STAT3 as well. So our kind of central hypothesis was that the inhibition of the JAK-STAT pathway could potentially be a therapeutic target for CTCL, and in addition that the JAK-STAT pathway mediates development of resistance to romidepsin. So our first aim initially was to test 6 JAK inhibitors in these 3 CTCL cell lines. We used JAK1 and 2 inhibitors, ruxolitinib, momelotinib, baricitinib, and JAK1/3 inhibitor tofacitinib, and then these JAK3 inhibitors, which are still undergoing investigation. And we found that the JAK1 and 2 inhibitors, specifically momelotinib and ruxolitinib, had the most consistent and potent cytotoxic effects across all the 3 cell lines with IC50s in the micromolar range. Given the results from the JAK inhibitors as single therapies, we next wanted to test whether romidepsin in combination with the 2 most effective JAK inhibitors, which were momelotinib and ruxolitinib, could have synergistic effects in cell lines. We found that the combination of the 2 had an additive effect. and that these results, while HUD78 is shown, were consistent across the two other CTCL cell lines. So next, since with this results, our next aim was to characterize the effects of JAK inhibitors in patient samples, specifically those that had developed resistance to romidepsin, and to assess whether combination treatment with JAK inhibitor and romidepsin could overcome this resistance and induce sensitivity again. So we had PBMCs were isolated from two Sézary syndrome patients who had developed resistance to romidepsin. These samples were activated for 3 days, and then we treated them with either romidepsin alone, JAK inhibitor alone, either momelotinib or ruxolitinib, and then combination of the JAK inhibitor with romidepsin. Then we assessed apoptosis at 3 days, and then stat phosphorylation at 24 hours. In the first patient, the romidepsin-resistant patient, on the left we have the quantification of apoptosis, and then representative plots on the right. First, want to highlight that in the blue, that's romidepsin treatment, as expected, we saw that there was no statistically significant difference between romidepsin and vehicle treatment. We saw moderately increased apoptosis with the JAK inhibitor treatment, which is highlighted in red and green, and then importantly, the combination of the JAK inhibitor, which are highlighted in purple and orange, had significantly greater effect than any individual agent alone, and actually overcomes the romidepsin resistance and seems to sensitize these samples. We then had our second patient sample, and we see similar results as well. Romidepsin alone does not appear to have any statistically significant effect. We see moderate increase in apoptosis with the JAK inhibitors as single agents, and then we see significantly more with the combination therapy. And then to assess what's the potential mechanism of the JAK inhibitor and overcoming the resistance, we looked at STAT5 activation via phosphoflow cytometry in both patients. We can see that in the green and the blue that momelotinib and ruxolitinib, as well as the combination treatments, results in significantly decreased STAT5 activation, and that romidepsin alone, as compared to the vehicle treatment, does not result in any change, which is consistent with our hypothesis that the JAK-STAT signaling pathway might be the mechanism by which patients develop this resistance phenotype. So in conclusion, we show that JAK inhibitors, they're a promising therapy for CTCL in both patient samples and cell lines. There's a differential effect among the JAK inhibitors, even amongst those that putatively are within the same class, which suggests that there needs to be further development of targeted therapies. And lastly, we show that inhibition of the JAK-STAT signaling pathway overcomes romidepsin resistance in these patient samples and might serve as a sensitizing agent for future combination therapy. So I'd like to thank Dr. Larissa Geskin and also Teresa Palomero for all their guidance and support over the past year, and as well as other members of the Cutaneous Oncology Center and laboratory as well. Thank you for your time.
Thank you. Any questions? Please come to the microphone. I have a question. I was surprised to see how much cell death you were getting from just ruxolitinib alone. So are these cells just, how much of an effect are you having on cell survival versus also proliferation just with the JAK inhibitors?
So what do you mean in the patient sample?
In the patient sample.
So we, given kind of the availability of the patient samples, we chose to do apoptosis. We haven't looked at cell viability, but given that these primary samples don't do very well in culture, there's always a great degree of cell death, and inevitably, even after a few days in culture, they don't really maintain very well.
Thank you. So that concludes our session, and Stu Chandler will make a couple of remarks to conclude our session. Thank you.
Thanks, everyone. I just wanted to, on behalf of Aclaris, thank everyone from the SID and the scientific committee and the annual meeting committees. I don't know if any of you have ever run or been involved in putting these meetings together, but it takes a tremendous amount of work. I want to thank Dr. Travers and Brian, Dr. Kim, and the presenters for giving us really a great presentation today. and hopefully look forward to seeing everyone next year in Scottsdale. I think we were going to call the meeting next year New Jack City, but we're going back to an old city, so we're going to have to come up with a different title for next year. But hope to see you there, and take care. Thanks.